Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we jump into this new episode, we'd like to take a moment to share some exciting news with you. We are working on a new and exciting project. We want to bring the Dr. GPCR ecosystem even closer in one place and make it available at the tip of your fingers. Soon, you'll be able to access GPCR-focused information faster, interact better, and advance your work more collaboratively. If you are as excited as we are, you can sign up to be an ecosystem beta tester. All you have to do is go to drgpcr.com/ecosystem and enter your information. Stay tuned. More details will be released soon. Make sure that you mark your calendar for the third edition of the Dr. GPCR Summit. This year, the summit will be held between October 10th and 16th. Stay tuned, as we are also working on the program for the summit. Visit drgpcr.com to find out more details about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and I'm delighted today to have with us Dylan Eiger. He is one of the awardees of the Summit 2021 of Dr. GPCR Summit. Dylan gave a fantastic talk, and I'm so excited to have him with us today. Hey, Dylan.
Hey, Yamina. Thanks for having me on today.
Thank you so much for being here. Congrats again on winning one of the awards at the Summit for your presentation.
Yeah, thank you. And thank you so much for putting the summit on. It was really great to not only give a talk, but hear what other people are doing in the field.
Fantastic. Thank you so much. And we're excited for the next one coming up this year. But let's jump right into it. Would you please give us a little bit of an elevator pitch of who you are and how you got into working on GPCRs?
Yeah, sure. So like you said, my name is Dylan Eiger. I'm currently a sixth year MD Ph.D. student at Duke University. I'm originally from Philadelphia, Pennsylvania, but I went down to Duke to do my undergraduate education where I got a degree in chemistry. And originally I was actually studying polymer chemistry and material science, so really nothing related to living systems. But then I developed a passion for medicine and ended up applying to MD PhD programs and chose to stay at Duke for that level of training. And throughout my medical training, I really became interested in pharmacology, primarily in the cardiovascular system. And as I was learning more about the different labs that I could join here at Duke and what we're strong in, the GPCR field just seemed like a natural place for me to join. So I didn't really expect that I would end up doing receptor biology, but it's definitely been a great opportunity to not only study something scientifically, but something that's really translatable to actual patient care.
That's fantastic. A chemist. So we have a chemist, ladies and gentlemen, among us. That is awesome. Did you once you decided to go into the MD PhD program, and you were exploring the different lab options that you had at Duke, and obviously at Duke, you have a panel of fantastic labs that you could have chosen. Did you spend some time in each of these labs, or how did you go about choosing your lab?
Yeah, that's a great question. So originally I was actually thinking of just kind of extending off of what I did in my undergraduate work. So something in biomedical engineering, doing something more with biomaterials or tissue regeneration. That was actually my original interest. But before you do your PhD, you actually get some of your medical training done. So at Duke, we do about 2/3 of your medical training prior to actually doing your PhD. So that 2/3 of medical school involves the didactic years where you're in the classroom. But then it also involves a really significant clinical portion where you're actually going through rotations in the hospital, working with attendings, nurses, physician assistants, and seeing how medicine's actually delivered. So during that time, I got to meet a lot of different physicians and physician scientists who are doing translational work. And that experience in the hospital really shifted my interest away from biomaterials and more towards pharmacology. So at the end of the beginning of my medical training, and as I transitioned to my PhD, the PhD aspect of our program, we go through a series of lab rotations. So what's nice is in the MD PhD community, you don't affiliate with a specific department. So you could do these rotations in biochemistry or genetics, really whatever department the MD PhD program will let you affiliate with. So I did a rotation in the Department of Biochemistry. I also did do one in biomedical engineering, and also did one in pharmacology and cancer biology. And at the end of the day, I was really looking for not only a lab that interested me scientifically, but really the mentorship was something that I wanted. Just because the MD PhD is a little bit of a niche pathway, there aren't too many of us out there. So you want to affiliate yourself with people who understand the kind of two roles you play as both a physician and scientist, but also how they intersect in both the lab and in the hospital. So what I found was the GPCR community at Duke is really a mix of both scientists and physician scientists. And there are some just straight up physicians who we collaborate with as well. So while it is, I think, scientifically extremely interesting, the translational aspect of it was obvious to me. And the mentorship, not only from my principal investigator, Sudarshan Rajagopal, but also from, the myriad of other GPCR folks at Duke who are running their own labs.
That's fantastic. Let's take a total step back. You started in chemistry, you decided to go into an MD PhD program at 15 or maybe at 10. What did you want to do when you would grow up?
Oh man, it's a great question. So both of my parents are actually in healthcare. So my father is a physician and he specializes in pulmonary and critical care, and my mother's a nurse. But my mom is from the Philippines and my dad is from New York. And I got to say, growing up, one of the things that I loved about our family was just how diverse the food was. So my mom, Filipino culture has lots of different types of food, and my dad's from New York, and that has its own culture. So I really loved food growing up, and deep down, I think, and I probably still want to, like something with food or wine, like that has always been a passion. I did have to ground myself then and say, okay, what am I actually going to pursue at the end of the day? So when I entered college, following the pre-medicine track was really what I defaulted to, but I wasn't set on that by any means. I'd never done any form of research before I arrived at Duke for my undergraduate training. So when I got in a lab, it was primarily just to figure out if this is something I want to do or not. Not necessarily that I came in with this interest of, being an engineer and doing polymer chemistry and material science is what I wanted to do. I actually just found one of my chemistry professors who was a great mentor. And I worked with him simply because I liked how he taught and how he made me think. So even though I had this developing interest in medicine, I started to learn more about the research field. And then I honestly still was just like, okay, I'm probably still going to go to medical school, until one day I was at a barbecue and I ran into the actual MD PhD director at Duke. And that's when I learned about MD PhD programs. And I learned that was exactly what I was looking for. A combination of both scientific work and clinical training, and finding the intersection between the two. So I really didn't figure this out until late in college. So when I was younger, I probably would have said a doctor, probably actually wanted to be a chef. And sometimes I still feel like I would want to be a chef, but my cooking skills are not very good. I love eating, but cooking, I'm still working on.
Well, weekend culinary school is still not out of the question then.
I know. I know. That's something I definitely need to set aside some time for.
Well, when you think about it, you're still cooking. You cannot eat what you're cooking, but you're making something in the lab. So that's
exactly. There's a lot of similarities, following a protocol is like following a recipe, and there are some do's and don'ts. So maybe that's why I ended up where I am. It's just an extension of cooking.
Perhaps it's the scientific mind and the cooking combined. That's where the lab might come from.
Yeah, possibly.
That's so awesome. And as you mentioned, GPCRs and being in the clinic and having those classes, those pre-med classes, the training in the hospitals. But where do GPCRs enter the equation for you? Was there a moment where you said, this is what I want to do because this system is so interesting?
So I think when I realized that this was the field I want to do my PhD in was actually we have an annual MD PhD retreat. And when I was there one year, my current principal investigator, Sudar, he actually gave one of our talks. He was one of our invited faculty members. And I remember listening to him talk, and he showed me just the type of work that has been done at Duke, how translatable our work is, with about 1/3 of all the drugs targeting GPCRs and how they're implicated in almost every physiologic and pathophysiologic process. But also just seeing the translational aspect of it was really important for me to see that, you've got tons of people at Duke like Sudar, like Bob Lefkowitz, Howard Rockman, all these physician scientists who study basic biochemistry in the lab, then are also treating patients and using different drugs that target these receptors. That to me just seemed like a really exciting and interesting area to be in, because you can really use both of your skill sets, the clinical side and the scientific side, together and on a day-to-day basis. I didn't feel as though I had to silo myself of, I'm going to do this specific type of medicine and then this really niche area of science that may or may not talk to each other. And then I think the other thing was just how widely applicable GPCRs are to every organ system. That was it was one of the recurring themes I found during my clinical year, was whether you're doing a psychiatric rotation or you're doing internal medicine or even surgery. I mean, these receptors show up everywhere. So just the ubiquity of their clinical utility was really, it kind of just said, okay, this is a good place to work.
I love how you're putting together, the clinical aspect with the biochemistry, and it is in my mind, it's a continuation, it's a process. We identify a receptor or family of receptors. We want to understand how these work. Once we know what the problem is in a pathophysiological setting, we want to target them, and then obviously the goal is to treat patients.
Right.
And it sounds like it is a nice cycle.
Yeah. And I think one of the coolest things, I mean, when I started in the lab, I was very naive to both medicine and science, and I still am, but it was very cool to be tested or just learn about these drugs in the clinic and feel like you have an understanding of them. And then when you get to the lab and you actually learn, we really don't know how a lot of these drugs work, and there's a lot more nuance to them. Then it got me really excited, because it says, even with our limited understanding, we have such great drugs available. And we're just scratching the surface of their clinical utility. Once we actually understand how these drugs work, then we can, the sky's the limit.
I've always found it mind-boggling that these drugs have, they're good enough, they're safe enough. We give them to patients, and doctors give them to prescribe them and give them to patients. These help patients, but when, exactly as you mentioned, when you go back and you look at the details of the pharmacology, of the biochemistry, you're like, oh my God, why would you even prescribe this?
As I've learned more about the GPCR system and just in the endogenous GPCRs and the endogenous GPCR ligands, I'm like, man, how do we not combust like all the time and just devolve into a pile of mush? And when you just learn how complicated this system is, it is pretty remarkable.
I think so too. And then, as you mentioned, there is always a GPCR involved. Anything you can think about, there's always somehow, somewhere, a GPCR or a family of GPCRs involved in that. But I find myself sometimes, people, when I talk to people from different fields, everyone is really focused on their own little field, their molecule, their proteins. And in my mind, I'm like, there's a GPCR involved. So you can work on anything you'd like. There is a GPCR involved. You may not be interested in it, but there is.
Exactly. Yeah, I got to say, not only from just the receptor field, but also a lot of the methods we use that have been developed in the GPCR space are so translatable to other fields of research too, that I think that's been something that's helped me as a trainee to just not only study our receptor, but also the methods here and find how well that translates to other areas of science. It honestly, I think it's a really great place for trainees, both intellectually and just professionally.
I think you make a great point about the translatability of the methods. Because when I think about, my discussion with Bob Lefkowitz or Paul Insel, when they were mentioning, there was a concept of an idea of a receptor, but nobody had purified it. Nobody knew the sequence. And look now what we can do. But also we've developed so many tools that can be put into other systems and to study other proteins that for now remain not as well characterized as GPCRs. And we also have to acknowledge that there's so much more to do with GPCRs.
Right. Yeah, I think you're spot on. And I think the one of the biggest examples, I guess, is the structural revolution in GPCRs. When we just look at the past decade, the number of structures that have come out, I honestly have trouble keeping up with it because of cryo-EM and the number of structures that come out. But I'm just floored at how fast our field moves. And I know you, like I, have done a lot of resonance energy transfer work, and that I have friends asking me now when they want to study how two things interact, they come to me because of how ubiquitous our technology can be.
Agreed. And then it's funny because now when I think about structures, I'm like, I don't know how many there are. I don't know them. GPCR DB, and you just look those up per receptor, and then you have all of those very nicely compiled.
Exactly.
All right, let's deep dive. And I asked this from everyone. What is your favorite GPCR?
Well, I'm biased because I study one, but it's a model. So I study the chemokine receptor, CXCR3. And the chemokine system is a system of both receptors and ligands that are found endogenously. And it's a very promiscuous system in that a lot of ligands can bind to multiple receptors and vice versa. One receptor could have multiple ligands. So as and a lot of the listeners will know, is that this phenomenon of biased agonism being really studied in the GPCR field. We in the Rajagopal lab are really interested in an endogenous example of bias. So we use this chemo system really as just a model. And CXCR3 has been our receptor of choice. It's involved in a lot of different diseases, cancer, cardiovascular disease, shows up in COVID all of the time whenever someone does some big omics screen. But that's my favorite. There are some also, I guess the second place are the atypical chemokine receptors. So I guess still within the same field, because they exhibit some very weird and non-canonical signaling or behavior that most GPCRs don't show. But I got to go with CXCR3, mostly because I've spent 4 years on it, and it would feel bad if at the end I came out with a bitter taste.
Yeah, you'd say, oh, I don't know, a beta adrenergic receptor, or some funky receptor. Well, you must probably know that I also worked for a long time on chemokine receptors, including CXCR3. And I think that was my Everest when I think about working with chemokine receptors, because during my postdoc in Tom Sackmar's lab at Rockefeller, my idea was, CXCR7, which is one of the atypical chemokine receptors, heterodimerizes with CXCR4, but CXCR7 also binds CXCL11, which is a ligand of CXCR3. And the idea was, let's just test if CXCR3 and 7 interact. And then Tom asked me this very interesting question. He said, well, you have 3 splice variants of CXCR3. What do they do?
Right.
And I spent 5 years trying to figure out what do they do.
Right.
And it was a long 5 years, but it ended up being such a fun project to work on. So I can definitely relate to your love for CXCR3.
Right. No, I know exactly what work you're talking about. We just submitted a manuscript and I've cited it. So I've read that paper. Don't worry.
Can't wait to read yours.
Yeah, thank you.
So I know that you mentioned that CXCR3 is a model receptor that you're using in the lab, but what is the status of the research with CXCR3? Like, in what context does the information regarding CXCR3's signaling, and in the context of bias signaling, what does that give us as information, not only for this receptor, but for the field in general?
Yeah, great question. So there's really, I'd say, 2 main reasons that I'm interested in studying it. One is, how can we better understand bias, not only at the chemokine receptors, but broadly across all GPCRs? The second, which is a little bit more of a clinical goal, is we really don't target this system. I think primarily because it's so promiscuous and originally was thought to be like a pretty sloppy target, and that you're going to have so many off-target effects. But I really think because it shows up in so many different inflammatory diseases that it would be a prime candidate. So in terms of where we're at with our research, and really what my doctoral work is focused on, are figuring out mechanisms of bias. And there are a ton. I think it all starts with forming an ensemble of receptor conformations that bias signaling through some effectors over others. But really, what else can happen? So 2 of the projects that I've been interested in are one which I presented at the summit, looking at differential receptor phosphorylation, of the CXCR3 C terminus. So this idea that 3 different ligands can all bind to the same receptor, and that leads to differential phosphorylation of that receptor, which then leads to differential engagement of effectors, most notably beta-arrestin. So that's been one of the main projects that we've worked on. And I think what we've come to show is not only do we now have a lot of evidence that biased ligands, endogenous biased ligands, can form different receptor phosphorylation barcodes, but in this work, we actually show that that does correlate to function, both from a signaling perspective and from cell-based assay, just looking at T-cell migration. So it's showing that if you can target these receptors and figure out which barcodes lead to the types of signaling that you want, could you potentially, I mean, this is speaking in an ideal world, could you find out the patterns which are therapeutic and those that are pathologic, and try to make drugs which bias the signaling in that direction over another? The other project that I think came out of using CXCR3 as a model is this really, I think, like a new exciting area of GPCR biology or biochemistry, which is subcellular or location-based signaling. So understanding how a receptor, we normally think of GPCRs being at the cell membrane, but knowing that they exist on a variety of different intracellular membranes, and they can also traffic. And there's been a lot of great work in the past few years. A lot of it's out of Mark Van Zostro's lab. We have Nina Svetsanova at Duke, who's a postdoc with Mark, who's doing some really great work studying GPCR translocation and subcellular signaling. So that's one of the other projects we've worked on, showing that you can get a biased profile, a biased signaling profile at the cell membrane with the CXCR3 based off the ligand you use. But actually, as that receptor then traffics, the ligand or the bias signaling changes. So not only is there this idea of ligand bias, but we now show that there's location bias, and there's some intersection between the two, that not only does it depend on the ligand you use, but also where that receptor goes. So honestly, at the end of the day, I think I've probably created more questions than answers, which is, I guess, a good thing. But it just shows me that there are multiple mechanisms of bias, which I hope will translate into multiple different ways of targeting this system to get a therapeutic but specific pharmacologic effect.
I love it. And I do love the idea of bias and the concept, and love chemokines and chemokine receptors because it's such a complex system. And just being able to say, okay, this receptor with this chemokine is going to initiate this phosphorylation barcode on the C terminus, which is going to end up taking the receptor to this subcellular localization. And at the end of the day, this is the overall effect on the cells or overall effect on an organ, or, in a pathophysiological setting. This is the what I'm looking for the word, but this is definitely the core that we want to strike in order to, help the patients.
Right. I think it's definitely too reductive, but
I know, but yeah, I know, but then again,
right. I think it makes sense that because a single receptor can generate so many different signaling profiles, there has to be, ideally, I would like to believe that there's some proximal way that directs everything downstream. And I think it all definitely starts with the ligand receptor interface, and then the ensemble of conformations that the receptor makes. But then are there other, the ripple of effects that happen after that? And how does that specific ensemble of conformations engage different effectors? Can we actually target it downstream of that as well? But I agree. If we can, I know bias has been this like promise in our field, and last year we did have the first FDA approved biased agonist at GPCRs, but I think we're just scratching the surface because we don't fully understand how to actually manipulate this phenomenon.
Agreed. We're not there yet. Have you ever? So we know that receptors dimerize, and I'm not asking you to necessarily come up with the right answer. Let's just fantasize, because we're fantasizing about chemokines and chemokine receptors.
Sure.
One of the questions that every time came up during my Ph.D. was, okay, I was looking at CCR2, which is also a chemokine, and it binds these 7 different natural ligands. And the question that always came up is, do these chemokines dimerize? Or have you tested mixing the chemokines to see what the signaling looks like? So let me ask you the same question.
Okay. Oh man. So I'll be frank. When I started my PhD, I was like, the idea of receptor dimerization, I'm like, I don't know if I buy it. But I've definitely been converted, and I've seen a lot of really great evidence coming out. A lot of the reason I was so skeptical initially was because of a lot of our work is done in overexpression systems where everything's overexpressed. So how much of this is physiologically relevant versus just an artifact of experimentation? I have not personally looked at CXCR3 dimerization. Or with homodimerization or heterodimerization. If we've done anything, I'll tell you, it's primarily been looking at, like you mentioned, CXCL11 binding to CXCR3, but also binding to ACKR3 of CXCR7. Yeah. So that's where I've been focused. I have not looked at dimerization or heterodimerization, but I mean, I buy it. Again, it just adds complexity to the system that we still don't understand.
Exactly, and I think that's also that's another level of complexity. I think that should be explored, but you may not be able to do everything in during one PhD or one postdoc.
And you know, the dimerization question again, it's really hard because you know, I believe that these events happen, and now that they can alter signaling from each receptor. The question is really, you know, does this happen endogenously? And that's really what I would love to see. There is work coming out now showing it, but how much of this, you know, the promiscuity of the chemokine system, is it simply just the number of ligands, the number of receptors, the number of combinations you can get there, or how much of this role does dimerization play? That I'm honestly, I couldn't even take an accurate guess.
I don't either. I don't think anyone can give us, you know, the answer to that. But have you ever tried, not the receptors, but looking at the chemokines? Have you ever mixed, I don't know, CCL9 with CXCL9 with CXCL11, or you know, any, because that was so here is why I'm asking, because we're always talking about, you know, diversity and the fact that one receptor can bind multiple chemokines, but in a physiological system, you have all of these chemokines coming and going, and then, you know, to be honest, I've never done such an experiment because I had so much to do anyway with one chemokine and one receptor in these assays. But let me ask you, have you ever mixed these guys together?
Oh, I've thought about it, but I'm sure, as you know, chemokines are pretty expensive, and my boss would kill me if I started playing around with them and just haphazardly. But I have thought about it because you're right. That has to be the way that this exists endogenously. Whenever I see, again, omics papers come out, you know, whether it's RNA-seq or proteomics, I always see, generally, I won't say always, we'll see at least one or two CXCR3 chemokines show up together. Rarely do I ever just see one. It's primarily two I'll see show up together. So then it does beg the question, how does the receptor signal when there are two of these chemokines around? Is it quote unquote additive, or is it synergistic, or what is it? Or is it even antagonistic? I don't know. The other thing I'll say is, in that talk that I gave at the summit about the phosphorylation barcode, the CXCR3, what I'm finding is that sometimes even proximal signaling assays don't correlate really well with the physiologic response that you see. So one of the ligands of CXCR3 is CXCL9. And I honestly thought it was like a dud of a ligand, in that it doesn't really do much. It has really poor potency when you compare it to the other two ligands of CXCR3. Yeah. But when we study CXCL9 in our functional assays, it does a lot. And I don't really understand how. So this is a very long-winded answer, and I apologize, but the thought has been there. I've been terrified, one, from being reprimanded by my boss, but also because I have no idea how I would even interpret the data because of how complex it is.
I oftentimes thought about it as well. Like, what do? You keep one constant concentration of one chemokine, and then you do a dose response on the other, and vice versa. And then what does that tell you? You know,
it's just going to confuse you more, would be my guess.
I kind of agree on that one. And yes, I think CXCL9, if I remember correctly, CXCL11 did everything and turned on everything and anything. But then CXCL9, compared to the other two, as you mentioned, I think it, in my hands at least, it internalized beautifully. I believe it also recruited beta-arrestin, but there was no GI whatsoever
Yeah.
signaling.
It honestly seems like a partial agonist. But then when you, at least at proximal signaling assays, but then when you look downstream, I mean, it really can do a lot. And then, the other thing, we've done some omics studies on these three chemokines, and our data looks, what we see actually correlates really nicely what has been seen in vivo, where like CXCL10, it's a relatively G protein biased ligand. It induces this pro-inflammatory phenotype of T cells. And there's a nice paper showing that CXCL10 does lead to worse disease in this encephalomyelitis model. But then when you look at CXCL11, that one that you mentioned turns on everything, it seems to not do anything inflammatory. It does a little bit inflammatory, but it also seems to regulate cell cycle a little bit differently than CXCL10. So maybe it's involved in T cell growth. So how you get such divergent physiologic responses, honestly, I have no idea.
But still, I think it's beautiful. I think it's just such a cool data. And then it begs the question of, okay, what next? And how can we generate new tools, or what are the tools that we actually need to really dig deeper into these events, not only in this system, but in other chemokine systems or any other GPCR systems, basically.
Yeah, I completely agree. I think one of the things that I love about our field are all of these assays to assess signaling. Like, I think we're one of the best fields in that you have such a great toolkit to choose from. But I think one of the next steps is understanding how this all translates to a physiologic response. So taking a systems pharmacology approach, Mark Knepper is really, that's been his approach with the vasopressin receptor, has been doing big omics studies, trying to figure out how do we integrate all of these different signaling pathways into a physiologic output. I think we're not there yet. I don't know if many fields are there yet, to be honest, but I would really like to see that as one of the new kind of directions of our field. Is we've shown that bias exists. What does it actually mean, and how does it translate to a phenotype?
Agreed. And yes, I think it's also a combination of the different methodologies in that toolbox, and really putting kind of our ducks in a row and saying, okay, for this system, we're going to run it through a pipeline of these different possibilities that we can be measuring, and then you organize and you put this data potentially in a machine learning type of algorithm based learning system, and then something will come out of it, hopefully, where we can better understand at a higher level with a lot of data. And I'm thinking here, those responses in time course in different tissues, different cell types, hopefully endogenous levels of expression,
right?
And getting to better understand, getting an answer as to, okay, so overall, what happens?
Yeah. And I think I agree. Whenever the barrier to doing all that gets significantly lower, I think that's the next step. You're aware of this, but the opioid receptor was like the holy grail. And I still think there's a lot of promise there, but I don't think we understand the system as well as we should, because of all the things you mentioned, is the differences in mice, cell types, whose hands you're using. And I also think that, in our field, we really define bias as G protein versus arrestin. But there's been a lot of work showing, Andy Cruz had a really nice paper doing some proximity labeling on the beta-2 and the angiotensin receptor. And there are thousands of proteins that interact with GPCRs. So I sometimes wonder if our idea of bias is being limited to these two critical effectors of GPCRs, maybe some of the nuance actually comes from other effectors that we're not paying attention to.
You make a great point. And I think it also, the focus that we currently have on G proteins and beta-arrestins is not because of a preference, it's because of the tools that we've developed to look at these pathways.
Right.
Looking at G alpha 12, 13, all of these more complex, more finicky, more tricky pathways is not as well developed. Actually, it's getting much better now, but the tools that you can very quickly look at GI, GS, and beta-arrestin 1 and 2, and then that's it. But I'm pretty sure, I think I could totally agree with you that there's other things that happen.
Yeah.
Close to the plasma membrane, but then what happens after that? Do we look at ERK signaling, AKT, some other phosphorylation events that are initiated by GPCRs?
Hmm. I completely agree. And I think with time, as the methods get better and there's more hands and money, then we'll be able to start exploring some of these other areas that really haven't been touched as well, as, like you said, the classic G proteins in breast and 1 and 2. Well, I think give it time, people will start to branch out a bit more just to see what else is out there.
Agreed. Speaking of tools, if you had a magic wand and you could create a cool new GPCR signaling measuring tool, it's a tough one. I know, I know it's a tough one. But what would it be like? It doesn't have to be necessarily a new pathway, but is there something that you wish you would have right now, or you had a couple of years back, to help you do your work better or more efficiently? Better understand your system?
So, let me think. The first thing that comes to mind, because we've struggled with this question, has been one, how to better understand beta arrestin's conformation. Because we've seen that you can have two ligands, both recruit arrestin to a similar extent, albeit arrestin then and goes and does different things at those under those two ligands. And there was a lot of work has come out of Bob Lefkowitz's lab looking at these different core versus tail conformations that arrestin can adopt. So at the summit, I talked about some of the conformational data that I've looked at, basically using these 3 chemokines of CXCR3 and using some CXCR3 phosphorylation deficient mutants to study how does that impact arrestin conformation. And long story short, it does, both of them do, but in a way that's not additive, that it's the conformation very much depends on the specific combination of ligand and receptor. So that's great. It shows us that arrestin can adopt a lot of cool different conformations. The problem is I don't have a way of actually studying if that conformational status translates into anything meaningful. So the tool we would love to have is a way to actually put arrestin in a conformation of your choice. Now, I know people can do that kind of by lopping off the C terminus, and you basically force arrestin to be in that one conformation, but there's a lot more granularity to it that you're missing when you do that. So that would be the tool I would love. It wouldn't really help my life. It'd probably make it worse because I'd have a lot more work to do. But I guess from like an interest perspective, that would be the tool that I would want, because I think that's something that's critical. I mean, there's a lot of evidence out there showing arrestin conformation matters, but I don't have a really great, like a clean way to do it. Right now, the tools are pretty dirty.
I love that. Thank you for that suggestion. Maybe someone listening to this podcast episode might reach out to you and say, "Hey, I know what you should be doing."
Oh, I love that.
I think well, that's the whole goal. I think
right.
Speaking of arrestins, that's one thing that I've always had kind of trouble with, is that you measure beta arrestin recruitment, and then when you're not really familiar with the receptor and you want to test out things, you compare beta arrestin 1 versus beta arrestin 2. And in my hands, in my experience, typically you get the same profile, independently of which beta arrestin you're using. So if I could add something to that wish list, it would be really having the ability of determining what happened, what are the signaling pathways that differentiate beta arrestin 1 and beta arrestin 2. Yeah. Because if you're looking at receptor and beta arrestin interaction, again, I don't know of any papers or many papers that describe very big differences between arrestin 1 and arrestin 2. Mm-hmm. But what do these two proteins do? I know that beta arrestin 1 is involved in the beta-catenin signaling pathway, and it does other things that beta arrestin 2, but we don't really look at that enough.
Right. I completely agree. And without spoiling too much, there's a graduate student in our lab who's interested in that question. What are the differences at one receptor? How does arrestin 1 do something differently than arrestin 2? Not only are they different in their signaling, but their localization within the cell is obviously different. So I agree. I think a lot of us at Duke, and especially in our lab, we study arrestin 2 primarily. And I remember I asked my PI, I'm like, why? Like, why did we decide that? And he's just like, I don't know. It's kind of what we've done at Duke. But there is a lot more. And I completely agree that that's the isoform differences, not only within arrestins, but in G proteins as well. That needs to be studied too.
Agreed. And you make a fantastic and an important point. the differences with the G proteins as well, because G alpha S, for example, has a long and a short version. Right now, I don't remember the differences and where that difference in length is, but which one would you pick and why? And is one more expressed in specific tissues versus another one? We don't know.
Yeah, and I'll say there was our lab recently published a paper. It was co-first authored by Jeff Smith and Tom Pack, and Tom Pack was in Mark Caron's lab at Duke, looking at this interaction between G alpha I and beta arrestin. And we see that this G alpha I beta arrestin interaction happens at canonically GI coupled receptors, but it also happens at receptors that don't canonically couple to G alpha I. So when we were studying that paper or working on that paper, a lot of the work was done with G alpha I1. And I believe in one of the supplemental figures of that paper, if not somewhere in one of our lab notebooks, but we did look at if you compare G alpha I1 to 2 to 3 and their abilities to interact with beta arrestin, they're different. So that right there gave us a little bit of insight. Okay, well, how did G alpha I and berestin interact? And the fact that we see differences in their ability to interact may give us some structural insight as to how this interaction actually happens. So I hope I didn't give away someone's project in the lab right there, but I think that's something that is really important. Are the isoform differences within G and arrestin?
Those are all, I think, all valid questions, and I think there's going to be a couple of PhD students and postdocs who will have enough work to do to kind of try and answer those questions.
Yeah.
All right, super. So I typically have this question, and the question goes as follows: what is your advice for young scientists aspiring to contribute to the field? Now, we know that you're an MD PhD student. And let me rephrase that question, because a lot of times when I talk to people, they tell me, well, you know what, I haven't been in the field long enough to say anything and to be able to answer this question. So let me rephrase the question. What are the things that you've learned in the past 6 years about the field, and also about being a scientist and an MD medical student, that you feel like you could tell yourself, your younger self? What did you learn?
Yeah, it's a very good question. There's a lot of things I would have told myself 6 years ago. I would say the first thing is, at least I made this decision, and some people may disagree, and it's just difference of opinion, but I really chose my research field for something that interested me, both scientifically and clinically, but as I mentioned before, primarily for the mentorship. While the PhD is a time for you to definitely become an expert in a specific field and learn a really great set of skills, methods change with time, but learning how to think, I think, is really important. And while you could be in a lab that publishes really well, if you're not getting the training that you need to become a good scientist and someone who knows how to ask a question, ask the right question, troubleshoot data, or work through methods, that is a skill set that you can't just pick up from a book, that you want to be around both a boss or a principal investigator and other fellow lab members who are going to teach you those skills. So I would say that would be one thing that I would kind of tell myself, is to prioritize that. Yes, the intellectual curiosity for whatever you're studying needs to be there, but also the people that are going to help you become or develop into a better scientist or physician scientist, that also needs to be a big part of the equation. And then other things I'd probably think about are making sure what you're doing is relevant. And relevant is a subjective term. Because I'll say a lot of times I got this probably a bad reputation in our biochem seminars, where I would always be the guy who would say, okay, why do I care about this research? Other than just intellectual, just intellectual curiosity. Why should I care about what you're doing? And that was something that I really had to keep asking myself. Was there were a lot of times where there were scientific questions that I was really interested in. And that's during my PhD and back when I was a medical student, that I had to take a step back and say, okay, why am I asking this? Other than curiosity, is there a reason? Because at the end of the day, and my training is really to understand how science is done, how medicine's practiced and delivered, how those two talk to each other, and really kind of be that liaison between the two fields. So I wanted to make sure I was just doing something that, yes, interested me, but did have some utility, because at the end of the day, a lot of the work we do is for patients and for the betterment of improving healthcare and improving the tools we can use to just make people healthier. So that was something that I would try to emphasize, as well as think about what kind of difference do you want to make. It doesn't necessarily need to be in health or medicine. It could be in energy. It could be in anything. But seeing, taking a step back and saying, what kind of difference do I want to make? And what research questions are going to help me contribute to that goal of mine. So I'd say the mentorship, and then just big picture, what you want to accomplish, would be the two things I would focus on.
You mean, and these 2 points are just spot on. I think the mentorship question is just very important. As you mentioned, you need to be in an environment where you can make mistakes, you can learn from them, and it's not the end of the world if you make a mistake. It's a lesson learned. And having that expert mentorship, that support that you need to develop, is just, you can't go without it. And I love the fact that you said that you cannot learn it from a book. You have to do it, and you have to do it in an environment that allows you to make those mistakes and learn from them.
Yeah. I have to give a shout out to everyone at Duke. My PI, Sudar, the other members of our lab. Really, the GPCR community at Duke has been such a great environment to train in. Bob Lefkowitz, Mark Carone, Howard Rockman, Sudarshan Rajagopal, and we have a lot of other faculty that I'm not mentioning, but they have their doors wide open. I mean, the amount of collaboration that we have, just bouncing ideas off of one another. But I really love is that all the senior faculty members, their doors are open and they want to talk to you. But what I really like is a lot of the time they're brutally honest, because they all deep down want to make you a better scientist and better physician scientist. So they're going to help you do it, not sugarcoat things, and just work to make you a better investigator. So that was honestly probably the number one reason that I ended up entering the GPCR space, was to get that mentorship and training, because these are people that I really look up to. The science is great as well. Don't get me wrong. I really like it, but it was really the combination of those two, which sold me on joining this field.
I love that. And I think a lot of listeners who are, contemplating going for a postdoc or contemplating going to grad school for a Ph.D., can very much use the advice that you just gave them, because yes, I agree. Mentorship, very important. You need to pick the right lab. You need to pick the right mentor. And also, yes, you do have to ask yourself, and then so what?
Right.
Like what's, I feel like whenever we're working in the lab and we have our own little project, it's important to take a step back and answer that so what question. And sometimes when, things are not working, you feel like you're just doing something and nobody cares. It is not true. I see this as a grain of sand in the desert, and we're just adding to that desert or to that sand in order to better understand this particular field, the GPCR field, and ultimately improve human health.
Yeah, it's a really nice way of putting it, because you don't want to think that your research is insignificant. I mean, at the end of the day, you can always find a reason as to why what you're doing is important. But I have to put a plug for the GPCR field. It's so nice to be around people who you can talk to all the time. And we kind of all realize like what we do is clearly has implications in human health. So it is nice to have that there just all the time as a nice reminder of why we do the work we do.
Agreed. All right, let's close up with the last two questions I have for you.
Right.
The aha moments. Any aha moments as a scientist while you were working on CXCR3?
Oh man.
Many?
I'm just, I'm trying to push all the failures aside and see all this.
Well, it's interesting because I think you shouldn't push the failures aside, and you should tell us a little bit more about the failures, because one, it makes people understand that failure happens and it's okay. Two, and I think it shows strength of character for everyone in the field who goes through 90% of failures, and then that 10% makes us go back to the lab, basically. Yeah. And I don't know if you remember those.
Oh, I remember my failures vividly. They've taught me more than the successes have. But one failure, which was my naivete with this field, was we were studying trying to look at downstream effects of CXCR3 signaling. And we found this protein that seemed, whose expression levels changed in a biased fashion. So we followed this up, got some data on it. And then about 6 months into this project, someone looked at me and was like, hey, Dylan, the molecular weight of this protein is just not what's predicted. And I was like, yeah, that is a little funny. I never really thought about that because it's just an antibody we bought from a company. Long story short, this protein that we were studying, because of some artifact of the way proteins were named, there's essentially two proteins with the same name. So we were using an antibody and blotting for the complete wrong protein this entire time. And I remember that being like such a crushing day because I was like, oh my gosh, I just spent 6 months studying this thing. I don't even know what I'm studying now. And I remember just leaving feeling so defeated. And I talked to my PI, Sudar, and he was like, this stuff happens. Like there have been multiple instances and funny stories at Duke where people have just followed the complete wrong thing and they found something, but you kind of just have to roll with the punches because stuff happens like this. But what that taught me was, okay, you need to be a little bit more diligent and attentive to what you're doing. And that was one of probably the aha moments of have a good lab notebook, like simple things I probably should have known. But I have no shame in admitting that stuff happened to me because it made me a better scientist and who I am today. And then I actually think the other thing that happened was I was at one of my committee meetings, and Bob Lefkowitz is on my committee. And at the end, I presented all the work I was doing, and then I also briefly mentioned a few other projects I was working on the side. And after the meeting, Bob just wrote as my feedback, it was just focus, focus, focus. That was all he wrote. And he goes, Dylan, I really like your enthusiasm. You clearly have a lot of passion for this, but you are never going to get anything you want done if you do not focus. And that was some of the best advice I got. It was very simple. But after he told me that, and he just told me how to direct my thinking, I was able to be much more productive than I was previously. So I actually put a lot of projects on hold and I just focused on 2 and I was able to push those out. So just, I guess, being attentive to detail, knowing that things are going to happen that may or may not be out of your control, but then focusing, finding a project that seems promising, failing fast, learning from failure, and not being afraid to ask for help. That was something that I quickly became comfortable with, is recognizing, okay, I could sit here and try to learn how to do this thing, or I could just recognize I don't know it and go ask someone who does, and we can get through this a lot faster. So
agreed. You make a fantastic point, and you kind of alluded to it when we were talking just a couple of minutes ago before I asked this question about not only the focus, but about the fact that you have to ask the question, so what? What does this mean? And the thought I had while you were answering it was, well, we have a limited number of hours in the day.
Right.
And we have to focus on what is the most important, not necessarily what we feel like is the most fun to do, although ideally you want it to be fun and important,
Right.
But you have to figure out where you want to go. And once you have that objective, you have to, as Bob said, focus, focus, focus.
Yeah.
And it comes with challenges, with ups and downs, but doing something to do something might not be the best approach.
Right. Yeah. And that's 100% something I've had to learn. If I have these weird ideas of experiments I want to do, I'll write them down. And then the first question I ask myself is, 1, am I actually able to do this? And 2, can I do it quickly? Or is this going to take me a few months? It's going to take me a lot of time. I then take a step back and say, is this important? Like, is this important to my personal development towards my Ph.D.? Is this important for the field? So I've gotten better at assessing those questions and answering them. But that just takes time and experience. And the way you get good at it is by failing and getting this feedback from people that you very much respect, who have been through this and have trained tons and tons of people. They're going to know if you're heading in the right or wrong direction.
Agreed. And I wish I knew this, or somebody had told me this during my PhD. But you learn and you gain confidence, and it's just by failing, and you just have to do it.
Right.
At some point, I think, thinking about something and hoping for it, or thinking, oh, I'm going to do it, and maybe doing it halfway. No, you have to figure out, can I do it? Does it make sense to do it? How long is it going to take me to do it? But I think the most important question for me was, is there anyone who can do it better than I do? Because they're doing this day in and day out.
Yeah. Yeah. And I think at the beginning, I remember I was really floundering. I would get to lab, I wouldn't feel comfortable with what I was doing. So my boss, Sudar, was very much like, okay, in the beginning, you and I are going to have really short meetings early on. I know every principal investigator can't afford that to their students, but I would highly suggest in the beginning, really short, quick check-ins. And that's something that we actually do pretty frequently now. It's this thing called Scrum, where we'll have a short 5 to 10 minute meeting 3 times a week, just to be like, what's going on? Are people struggling? What can we address now rather than let it linger? But to get back to the story, he would meet with me every day for a few minutes just to see what I was doing. And that was so helpful because he was able to catch me quickly when I was heading in the wrong direction, and make adjustments. And after a few weeks, if not maybe a few months of that, I felt comfortable of like, I know how to troubleshoot things and how to think through problems, because you and I could sit here saying, is this the right question to ask? But when you start in a lab, you have no idea. Like you don't know the literature well. I probably still don't know the literature as well as I should, but
I don't either.
You just got to ask people and not be afraid to ask, and know that there are people who are, other people are such a great asset that sometimes we don't think of. And that's why kudos to you for putting together this network of GPCR researchers, because I'm so fortunate at Duke to have people down the hall who I can rely on. But if you're not at an institution that has so many GPCR focused labs, it can be tough. So I think the collaboration that we have at Duke, but will be improved on by the network of GPCR researchers you're making, is just going to accelerate our work.
You make a fantastic point, and yes, I agree with you. And I don't always have the time to follow the literature. I know that as a PhD student or as a postdoc, I spend more time at the bench than reading papers.
Right.
But one advantage that I can definitely tell you right now with Dr. GPCR and the podcast and meeting all these people who work in the field is that I know now that I don't have to know everything. I just have to know who to ask.
Exactly.
And that's such a great feeling. And hopefully we can translate that to more people in the field.
Yeah, I think that's probably, when you ask me the question about what method would I have had as to improve my work, I picked one that would make my life worse off because I'd have more work to do. But I mean, honestly, an easy way to collaborate with people, because there are so many different things you can do in our field, whether it's structure or imaging or breadth or, in vitro work or in vivo work. I mean, not one person can do it all. And if there's a quick and easy way to talk to others, that would be such an advantage.
Absolutely. Absolutely. So last question. When you're done with your PhD, you're an MD PhD, what's next for you, Dylan?
That's a very good question. I started to figure it out since I'm actually going to be defending my PhD in about 2 months. So I'll be heading back to medical school in May. I'll defend in March, but then I'll have some time off, finish up some lab work, go back to medical school in May, and then I have 1 more year. So after I get my degrees, I'm going to pursue a residency in internal medicine, but then plan on doing a fellowship in cardiology, just like my PI and a lot of other people at Duke. After that, I have absolutely no idea. I know during my residency and fellowship, I want to do research. I'd like to stay in the GPCR field. So wherever I end up for my clinical training, I'm hoping that there will be fields there or labs there that do GPCR work. But then after I do my residency and fellowship, I couldn't tell you whether I'll focus clinically, start a lab, do a combination of the two, drop everything and open up that restaurant I mentioned about earlier. Who knows? We'll see. But at least for now, it's to do the next level of my clinical training in medicine and cardiology.
Fantastic. Well, we wish you all the best. We'll think about you in March. I also defended my thesis, although it was almost 11 years ago in March. It's okay. March 31st, and it was my PI's birthday. And I kind of well, I don't think he's going to hear this, but I did a kind of cheat because there were 2 people that I needed to have on the committee, which was Michel Bivier and Thierry Hébert. And I told my boss, I said, this is the only day they're available, so we have to do it on his birthday. So his birthday coincides with my PhD thesis defense anniversary.
Oh, I like that strategy because you don't want to have your PI associate their birthday with a bad day. So, that was a good move on your part.
Yeah, absolutely. All right. Well, thank you so much, Dylan. I really appreciate it. I love talking to you. Congrats again on I guess submitting your PhD thesis. Good luck with everything, and obviously keep in touch. No matter where you end up for your residency, the GPCR field I think is always gonna be there in your home. Hopefully, we're diverse enough and we're dilute enough in the world in labs, so I'm pretty sure you're gonna end up working at some point in the GPCR lab in some capacity.
I hope so. And thank you again, Yamina, for having me on and putting together the summit. And it's really been an awesome opportunity to see this community grow. I remember when you first started out and getting the newsletters every month. It's really like a nice way to just stay connected to the community when we're, not only at our own institutions, but isolated a lot because of this pandemic. So, thank you for having me and for everything that you do to help connect us. And of course, stay in touch as well. And I wish you all the best.
Thank you so much. Thanks, Dylan. Bye.
Yeah.
Thank you for joining us and listening to this podcast episode. We'd like to thank our guests, as well as our team members, Attila Forrest and Inés Pinheiro. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcasts. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Stay tuned for the launch of our brand new project in the upcoming weeks, and email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.